Friends, in this year 2020, the Nobel Prize in Chemistry has been awarded jointly to Jennifer Doudna and Emmanuelle Charpentier. Dr. Emmanuelle Charpentier is currently the Director of Science of Pathogens Units in Max Planck Institute, and Dr. Jennifer Doudna is a professor in University of California, Berkeley. They have jointly received the award for their contribution to the development of genome editing method. This genome editing method is known as CRISPR-Cas9 genetic scissor. Friends, if we recollect the history of Nobel Prize, then we would see that in most of the cases the Nobel Award is given after 20 to 30 years of the discovery of that work. However, in this case, the first paper on genome editing was published in the year of 2011, and within 9 years of the discovery of this method, there are so many rapid applications of this method in clinical field that the Nobel Committee had to declare the award for this huge discovery in the year 2020. So from this we can have an idea that in near future this technique can bring a huge revolution for the mankind. So friends, let us understand about this genome editing process and how it actually works. Friends, before going into the details of genome editing, it is also very important to understand about the genes. Genes are similar to a binary code. The binary code contains information about a particular function. As you can see that this information remains as a binary code. Similarly, the genes also contain information about some particular function. However, the code that is used here was in terms of the sequence of different nucleobases present in DNA. These 4 nucleobases are adenine, guanine, thymine and cytosine. These 4 nucleobases jointly make this genome code in the same way as the two number 1 and 0 forms the binary code in different combinations. Genes are the basic molecular units of heredity. Many different features like eye shade color, skin color, hair type or color, etc. dependent on these genes. These genetic codes are generally carry forwarded from one generation to another generation. However, if we try to understand chemically what these genes are, then we will see that they are basically part of a huge molecule known as deoxyribonucleic acid or DNA. In this DNA, those 4 nucleobases are arranged in some particular sequence to form a code-like thing, and these codes are responsible to give instructions to some particular functions in our body. There can be varieties of functions for which these genes are responsible. As an example, how a particular protein can be made or how it can be made, etc. These proteins are responsible for the various types of functions in our body. So friends, let's see the exact location of these genes inside our cells. There are trillions of cells inside our body, and inside each of these cells there is nucleus which contains chromosome. This chromosome contains DNA, which is a large molecule. These DNAs are formed by 3 components. The first one is nucleobase. There are basically 4 types of nucleobases. As I have told you earlier, they are adenine, guanine, thymine and cytosine. These 4 nucleobases are abbreviated as A, G, T and C. In addition to these nucleobases, there are deoxyribose sugar and phosphate groups in DNA. All these components combine to form the nucleotide. The one particular sequence of these nucleotides which decides any particular function is known as gene. So genes are basically one particular sequence of those nucleotides. So friends, let's discuss about the genome editing. In the year 2011, scientist Jennifer Doudna and Emmanuelle Charpentier has discovered a new methodology which is known as genome editing. This method of genome editing is also known as CRISPR-Cas9. By using this methodology, scientists can make a very precise change in DNA to cure so many genetic diseases. The interesting fact is that this novel methodology has been discovered accidentally while they are working in different projects. The main aim of that project was to know how some bacteria fight against some viral infections. These viral infections are like ticking time bomb. The bacterium has a few minutes of time to diffuse it, otherwise they will die. So to fight against these viruses, there is already an immune system installed inside the bacterial cell, which is known as CRISPR system. This CRISPR system will identify that viral DNA and destroy it. This CRISPR system consists one protein, which is known as Cas9 protein. This protein can easily cut the double helical structure of DNA like a scissor. So basically, the research groups under the supervision of Dr. Jennifer Doudna and Emanuela Charpentier were mainly focused to understand the working principle of this Cas9 protein. The outcome of the research reveals the fact that the function of the Cas9 protein can be used in genetic engineering, which can make a huge change to the mankind. Using this genetic engineering, scientists can cut a particular portion of DNA and can replace it with others. This process can be done with a great degree of precision using the CRISPR technique. By using this CRISPR technology, we can achieve so many things that were never done before. This CRISPR technology has been already used on mice and monkeys to make changes in their DNA. Some Chinese scientists even showed that this CRISPR technology can be used to change the genes of human embryo. Some scientists of Philadelphia University showed that using this novel technology, one can cut the DNA of deadly HIV virus. So friends, there are lots of opportunities to use this genome editing technology for the treatment of many types of diseases. However, it raises concern about some ethical issues, as this technology can also be used in human embryo. So we have to be very careful that there should not be any misuse of this technology. So friends, let's understand how this CRISPR technology actually works. When any virus attacks bacterial cell, then it injects its DNA into it. The CRISPR system remains inside the bacteria, identify that foreign DNA, and it gets plucked out of the virus and inserted in little bits into the chromosome DNA of the bacterium. These integrated bits of viral DNA gets inserted at a site called CRISPR. Here CRISPR stands for clustered regularly interspaced short palindromic repeats. Basically, this CRISPR system is like a mechanism by which these bacteria are keeping a record that with time what are the viruses by which they got infected. And the important fact is that these informations are carried forwarded from one generation to another. So due to this mechanism, the bacterial cell become immune to that viruses for many generations. So this CRISPR methodology is like a vaccination card for the bacterium. When the bits of DNA of the virus is being inserted into the bacterial chromosome, then the bacterial cell produces a molecule which is named as RNA, which you can see here as orange in color. This RNA molecule is an exact copy of the viral DNA. RNA is also called as the chemical cousin of DNA. This RNA will interact with those entire DNA whose chemical sequence match with the sequence of it. So the RNA situated at the CRISPR locus also binds with a protein which is named as Cas9 protein, which you can show here as white in color. So this RNA and Cas9 protein jointly forms a complex and act as a guard inside the bacterial cell. The system always scans the chemical sequence of any foreign DNA, and whenever it matches with that of RNA, then this complex attached with that DNA. Here the protein Cas9 is acting like a scissor which cuts the viral DNA at the precise location, so the double helical structure of the DNA gets broken completely. Friends, however, the most important point is that this RNA Cas9 complex is programmable. The RNA, which is a part of the complex, is also known as guide RNA, and by a very small change in the sequence of the guide RNA, one can program the complex in such a way that it will identify some particular DNA having some particular sequence, and the complex will cut that particular sites of those DNA. This process is also known as genome engineering. By using this technology, one can do a very precise change in DNA. This is a type of repair process which is done in Microsoft Word. When we do some typo mistake, it automatically gets corrected. Friends, our cell can detect the broken DNA and repair it accordingly. So whenever there is a break in the double-stranded DNA, the repair process can be of two types. In one process, the two cut portions were stitched together, and in this process there will be a very minor change in the sequence at that cut position, and it will causes mutation. This process is known as disruption. In second type of repair process, the two parts of the broken DNA are joined by another piece of DNA from outside. This process is known as incorporation. Friends, we can understand it with a simple analogy. When our clothes gets torn, then it can be fixed in two ways. Either we can stitch the two torn parts of the clothes together, which is like disruption process, or we can take an extra small piece of cloth and join the two torn part of the cloth with it, which is just like insertion method. This is the way how our cell repairs the damaged DNA. So if we could break some particular location of our DNA using the CRISPR technique, then we could force our cell to repair it. So this repair process will be done either by disruption or insertion. Now as we know that in insertion process some other DNA is being used, so some new genetic information has been added in that damaged DNA after these types of repair process. So by creating these types of artificial defects in DNA using CRISPR method, we could enable our cell to repair for many defects or mutation which are responsible for different types of genetic diseases. Friends, this genetic engineering is not a totally new technique. This technique has been developed since 1970s. However, in earlier time, this technology was either not that much efficient or very difficult for practical applications. However, the CRISPR method developed by Jennifer Doudna and Emmanuela Charpentier not only simple but also very precise. Right now, scientists across the globe are trying hard to apply this technology to cure many human diseases. Generally, these experiments are conducted on mice and monkeys, as their DNA sequences are pretty much similar to human species. Here, as you can see that for the black pigmentation of this mice, some particular genes or DNA is responsible. By using this CRISPR technique, scientist has achieved to make a small change in that particular DNA which is responsible for this black pigmentation to produce these kinds of mice with white pigmentation. So as you can see, both these types of mice are absolutely same except a very tiny change in the DNA which is achieved using this CRISPR based genetic editing technique. So this CRISPR technique is so precise that we can make a change at the exact location of any DNA. So in near future, there are so many possibilities to apply this technique in different clinical field to cure many genetic diseases. Friends, this CRISPR technology not only can be used for the treatment of diseases but also can enhance human body systems. This technique can be used to enhance the strength of our bones, muscle or eye power. Moreover, one can get many desirable features like different color of eye shade, tall height, etc. using this genetic engineering. These features can make designers human in near future. However, till now scientists have very little knowledge that which genes are actually responsible for these types of features. However, in near future, if we can ever able to have information about those genes, we can practically make designers human being using the CRISPR technology. So friends, how do you like this video? Please comment down below. If you really like this video, then you can subscribe to my YouTube channel Chembiosis to get these types of interesting scientific contents. Thank you very much for watching it till the end. Bye. See you soon.